Well, good afternoon and welcome to MedTech Crossroads. It is Friday, November 20th, the last Friday before Thanksgiving. Can you believe it? We're almost all the way through 2020. Just a quick reminder, there's not going to be a show on Black Friday. We assume you're all shopping virtually for the holidays, so we will not have a show next Friday. Just as a reminder, also, there will not be a show on Christmas Day or on New Year's Day. We're going to miss you guys, but we will not have a show on either of those days. In the middle of December, though, we will be having shows. We've got some great guests lined up for December. But today, we're going to sort of make our way out into the holidays. We're going to catch you up on the COVID news, the charts that we look at every day. We're also going to look at surviving the next step in device development. But what we're going to talk about is applicable to any project you run. So I hope that the ideas we talk about today, no matter what you're working on, are going to be applicable. We'll talk about them in the context of medical device and medtech development. But I hope that no matter what you're thinking about this holiday season, after you're digesting that turkey or whatever it is, that you will think about this and some of those problems that are nagging at the back of your mind, maybe this will help a little bit. So we're looking forward to getting into what's called the Cynefin framework. After that, we'll have time for some Q&A. And after that, off we go to Thanksgiving. I hope you guys have a great holiday next week. And again, we will not see you next Friday. But with that, we want to get into the COVID news for the day. We've taken to showing you these graphs every time, and you can see that the hospitalizations in the United States are going up. The first peak was the Northeast, and the second peak was largely the South. The third peak is the whole country. What's interesting is that the deaths, as we talked about with Dr. Levy, are not as high as they were the first time, which we're all very, very thankful for. And if you didn't hear the show with Dr. Levy last week, you owe it to yourself to check it out. He presented a boatload of information, very fair and balanced too, in terms of what we can and can't expect in coming days and weeks. So we enjoyed that greatly. That's the picture of the US. The picture in Michigan, kind of what you would expect. The current hospitalizations are now starting to reach where they were back in April. But the deaths remain, approximately half of what they were in April, but they continue to go up. So this is something that obviously is the reason we've started bringing back the COVID news because of what we're seeing here. From the perspective of statewide PPE and bed tracking, not a lot of gigantic changes to report here. Things are still running full, but there's not an indication of overwhelm quite yet. If we look at the CDC, I wanted to bring you this because we haven't done a detailed look at this for a little while. These are the excess deaths in the United States. It's a grim statistic, but if you look at these green bars, the green bars for January through November of 2020 represent the number of deaths in the United States per week. That number is roughly in the 55,000 per week. I'm sorry, there's not a scale bar there. Roughly 55,000 per week deaths from all causes, excluding COVID. And the blue data represents deaths from COVID. The orange line represents a statistically significant number per week. In other words, you see with the flu season, the orange line is usually higher. In the summer, it's usually lower. And so for a bar to be above the orange line means that we're looking at a statistically significant number of deaths, whether from all causes, which is the green bar, or from COVID, which is the blue bar. You can't really take the most recent 4 weeks as truth yet. They look low. That doesn't mean they will be when all the data comes in. But you can see that this initial spike and then the secondary spike of COVID deaths. You can also see these two peaks, which is when people talk about deaths from other causes, like Dr. Levy was talking about sort of a new opioid crisis that we're having right now from everybody staying home and a lot of despair and other things happening. Those deaths that you're seeing that are statistically significant above the line are perhaps byproducts of COVID and the other social implications. If you compare those back to the 2017-2018 flu season, you can see that those statistically significant bumps are roughly the size of the entire 2017-2018 flu season peak. And so You get a sort of a sense there as to where we've been in the past, some of the not direct implications of COVID, as well as the direct implications in those blue bars. So as we continue to follow this, hope that information is helpful. We're going to go straight on to our presentation today, and that we're calling Surviving the Next Step in Medical Device Development. Now, oh, you know what? I'm sorry. We're going to back up. We'll restart that segment. We do have a comment and a question here from Jay Rajput, who is on the line with us from Beaumont. And since we're talking COVID, and I'm sure he's got some numbers, we're going to let Jay go live here before we continue. Jay, you're live with us. Oh, you're muted.
Can you hear me okay?
Yeah, you're great.
Excellent. Gene, once again, thank you so much for doing this. Really appreciate it. Really quick, just wanted to come out and make an announcement about Beaumont's vaccine trial.
Yeah.
So we are running a SARS-CoV-2 vaccine candidate trial. It's AZD1222. You know, if you look up, you can see that it's AstraZeneca's trial, though AstraZeneca says that we should not be taking their name, but whatever. It's the AZD1222 trial. It's basically a replication deficient chimpanzee viral vector based, candidate that we're using. In essence, it's really a weakened version of the common cold, the adenovirus. So the trials are, I think they've already started, or they might be starting. I know that they were starting in November, but what I'm going to do is, in the Q&A, I'm just going to put a link to our trial. When you go on to the website, there's a form that you can fill out, put in your contact number and or email address. They will reach out. You folks will reach out to you, ask you a bunch of questions to see if you're the right candidate for the trial or not. And in case of anything, I'm also going to put my email address in the Q&A. So if anybody has questions, they can reach out to me directly, and or my supervisor or my boss, Dr. Richard Kennedy. I'm going to put his email address as well. So you guys can, go onto the website, sign up, you'll get a call, and take it from there. Thanks so much, Gene.
Jay, thank you. That's great. And so this trial is just getting started now at this point with AstraZeneca?
Correct.
And if folks want to get involved with it and be a subject of the trial, they can make contact with you here. We'll get that on a slide here. This is good breaking news, so we will get this on a slide for people to see those links. That's beaumont.org/COVID-trial, and we'll paste those on a slide here for folks to see for posterity.
Thanks so much, Gene.
Jay, thank you for calling in. Perfect timing. We appreciate it, sir. And in fact, just because that is sort of breaking stuff here, we are going to, for the sake of the video record, we're going to pop that up before we start with our presentation to make sure that we get that in front of all of you, because I don't know if you can all see the questions and answers that are out there, but our friend Jay Rajput at Beaumont has given us the contact for participation in their trial with AstraZeneca. We're going to put Jay's email there. He did give this other email that he would like, so we will place that down based on Jay's approval of that, and we're going to share that over here so that you guys can make sure that you can see that. These are the addresses that you would want to go to for participation in the trial at Beaumont with AstraZeneca for the vaccine. So beaumont.org/COVID-trial. Our friend Jay's email, as well as his boss, Richard Kennedy, which Jay indicated you could reach out at any of these places to get involved, and I'm sure they would appreciate having participants in that trial. So that's exactly why we're here, to make those kind of connections. Jay, thank you for reaching out and popping on the show. It's great to have a place where we can all get together for this. I will just take one moment and see if there's any other questions, comments. If someone has a question for Jay, it looks like his connection is good, so he could take a question about that as long as we're on the topic of COVID. Just raise your hand if you have a question. And not seeing any, would say thank you once again, Jay. We'll look forward to hearing the results of that. Hopefully you'll come back and share with us as we hear more later in that process of that trial. Next up, we're going to talk today about surviving the next step in medical device development. Make no mistake, our subject is medical device development, but we're going to be talking in terms that will be applicable in almost any project area that you work in. If you've ever been frustrated and you've felt like, this should work, but it's not working, chances are what we're going to talk about today is going to enter into that discussion. It's going to help you understand better why we run into some of the roadblocks that we run into in projects. And this applies in the space of medical device development, all the way from prototyping to design documentation and testing, FDA regulatory filings, manufacturing. The principles we're going to talk about today apply in all of these places and all of these areas. So it's not specific just to regulatory or just to engineering. It's all over the place. Because what frequently happens is I think people are doing a great job of finding good advisors. On the show, we've had accelerators and incubators. We've had community members who are talking about things. We've had folks who do stuff like we do. And when we get together, you'll hear it. We're all talking about the same things, saying, oh, here's things that you've got to remember to do. You've got to remember to do this, and you've got to remember to do that. And so rarely are steps misunderstood. Usually you can figure out what is a step I need to do. I need a prototype, or yeah, I do need to be able to get to mass manufacturing, or I know I've got to get through FDA, so that's an important thing. I know it's there. But sometimes, frequently, there's a misunderstanding of inherent complexity. So next steps are often taken with the wrong inputs, usually too early or too late. Typical questions that folks will be asking, whether they're asking them out or not, they're saying them inside. Am I or my team, are we working hard enough to get my device developed? Could things be moving faster? I'm sitting here sort of trusting that things are moving forward, but could it be going faster? Should I invest more money? I'm an investor. Should I invest more money in this project, or are we just not hitting this like we should be? Should I hand out more tasks? Do I just need more bodies on this or not? Are we in a spread out and get more stuff done, or is this a 9 women can't make a baby in a month kind of a problem? What if I move too fast when I should have run more tests? What if I run tests and study this thing too much when I really should have been just getting it out there? When should I engage with the manufacturer? When should I engage with FDA? These are all questions at the front of people's minds. And how do you get answers to these? Well, the first thing to realize is that you may be missing a step. There may be a step that's missing in the middle somewhere there. And before you jump to conclusions and say, yeah, I've studied project management, I understand that there are things like dependencies, something that has to get done before something else. That's not what we're talking about. We're not talking about simple dependencies here. We're talking about something a little bit more complex. It's not just about dependencies. It's about known and unknown unknowns. Did you get that? We just used the word known 3 times. There are unknowns whenever we start a project that's worth doing. Some of those unknowns we know we're going to encounter. We've got to figure out an answer, but we know that it's coming, and it still requires work. Some of the unknowns are actually unknown. We didn't know that we didn't know. How do you can recognize that these things are there, but how do you address them in a meaningful way? How do you address them in a systematic way? Well, the first thing is to recognize that the world, while it is predictable and it is orderly in many ways, there are many ways in which it is not. It's all about understanding how predictable is the domain that I'm in. We know that the celestial bodies go around in a very predictable way. But what about the part of the project that I'm engaged in today? How predictable is that? What level of certainty can I assume in this? David Snowden and Mary Boone in Harvard Business Review in 2007 noted that all too often managers rely on common leadership approaches that work well in one set of circumstances but fall short in others. Why do these approaches fail when logic indicates they should prevail? The answer lies in a fundamental assumption of organizational theory and practice that a certain level of predictability and order exists in the world. Again, the world can be a very orderly and predictable place, but as we've learned in 2020, there's a bunch of places where it can go off the rails. And so the question becomes, for your project where you are, what's the level of predictability and order that's actually at the stage that you're at? That's what you want to understand. Well, fortunately, our friend Dave Snowden, not to be confused with Edward Snowden, Dave Snowden came up a number of years ago with what he calls the Cynefin framework. And you see one representation of that here. This is applied to all areas of project management. It's not specific to medical devices, but that's where we're applying it today. Dave notes that there are at least 4 or actually 5 domains. There's this clear or simple domain on the bottom right-hand corner. There's a complicated domain on the top right-hand corner. There's a complex domain on the top left-hand corner and a chaotic domain on the bottom left. In addition, in the middle, there's my favorite, the confused domain, which is right in between all of them and doesn't get you anywhere. We're going to talk about this, but I want you to know that you can go to cognitive-edge.com, which is where Dave Snowden has his body of work. He's got training there, all sorts of cool stuff. If you want to know more about Cynefin from a generic perspective, you can certainly go there. And the pronunciation of this is Cynefin, but it's written Cynefin, a Welsh word, I believe, and very confusing at first glance, but you pronounce it Cynefin. And Dave Snowden came up with this in 1999 when he was working for IBM Global Services, and it's really only making its way out into the mainstream now. But I hope you find it useful in your thinking. Let me give you an example. If you come to Into Being and you say, I've got a 3D model, it's one part, I'd like to prototype it in a polymer. Well, we can sit you down, we can ask you some straightforward questions. We can ask you, how strong does the material have to be? How hard does it have to be? Does it need to fluoresce or should it not fluoresce? What kind of a resolution do you need? If you can give us answers to those, we might be in a simple domain. And we could do stereolithography, or we could do fused deposition modeling, or we could injection mold a part for you. But what's interesting is that oftentimes when people come and we say, how strong does a part need to be? Well, they actually can't answer that directly. Usually they can tell a story about it. They can tell us a story about how the part will be used and the kind of people that'll use it and what conditions it'll be used. And from there, you can start to tell a story about the part where you could create a safe to fail experiment that would be to go on a discovery search for how strong that part needs to be. But do you see how we've moved out of what would be considered a simple domain just by asking the question, how strong does the part need to be? To truly understand how strong it needs to be, we have to exit the simple domain and go to a more complex domain to find out the answer. I'm going to give you a different picture of the Cynefin framework here. I love this one by Martin Berg, based on a 3-dimensional interpretation by Rob England. And if you carry one thing into the Thanksgiving holiday, it'll be this image, and you'll be eating your turkey, and you'll see this, and you'll wish you'd never seen it before. But if you think about these layers and these heights and having to climb between them, if we look in the middle, there's the same confused domain that we remember from before. Bottom-most is now chaotic, bottom left is complex, top is complicated, and then top right is clear. Let's talk about each of these in order and how we progress from one to the other in the course of medical device development. Well, let's start in the confused domain. We all start here at some point. We're confused about what has to happen, and it's easy to make a few different mistakes. One is to assume that everything is simple. Well, that's not true. It's also a mistake to assume that everything is chaotic. We want to stay away from the chaotic regime. We have no desire to be in the chaotic domain. We can deal with complex and complicated and simple, and it's just a question of figuring out how to move between those. So the most important thing from confused is to not try to jump up to simple and not allow ourselves to jump over into chaotic and despair of finding a solution. There is a pathway, there is a process. And that starts not with the clear domain, not with a simple domain. That's where you can follow a recipe like a cookbook. Well, many times you can follow a recipe out of a cookbook, but for medical product development, there's not a recipe for your particular device. So we usually aren't there, but let's go to where we normally do start. And that's in the complex domain. This is all about concretely identifying the unknowns to turn them into knowns, or at least into knowable projects. If you make a project plan at this point, it can only see the next step in your process. It can't predict the duration or the success of that step. And this is an important point. But don't assume that just because you're in a complex domain having to figure out what your unknowns are, that you're automatically in a chaotic domain. You're not. If you just sit down and start saying, what are our unknowns and what tests can we do to find out what other unknowns we have, you can quickly come to grips with where you are. So moving from complex to complicated can actually be very, very efficient working with folks with expertise in the domain. And the job there is to get as quickly to the complicated domain as possible. And speaking of the complicated domain, up one level, this is where things are knowable, but still unknown. We haven't necessarily done the legwork to reduce these ideas and these questions down to their hard answers. But as we start to see what it'll take to make them known, we start to gain control of our project timeline. And only when the right unknowns are made known, and usually documented, that we can decide whether we're actually in the clear, that we've made it to the top and can start operating in a more predictable way. And this process of climbing this ladder is really the work of medical device development. We're going to talk about the specifics here in just a minute. Well, when you think you've gotten to the top, we said it's a mistake to think you're at the top. It's a mistake to think you're in the clear. It's a mistake to think you're in the simple domain when you start. Assume you're in the complex domain and work your way up very quickly if you can. But are you sure you're in the clear? The penalty, and this is very important, and one of the things I love about David Snowden's model, the penalty for erroneously assuming you're in a simple domain is usually chaos. If you think you've got it all figured out, that's the worst place to be when you don't actually have it all figured out, because you drop off that cliff and you're back into chaotic. And it takes a lot of energy to climb back up that ladder because things have now been knocked into chaos. Fortunately, if you do find yourself in chaos, or if you think you're in chaos, it really just takes an admission, a willingness to categorize the current unknowns and to probe them, to get back to complex, get back on the ladder, and climb back up. Fortunately, the more mature your company is, the more mature your process is, the easier that'll be. Hopefully, it's just a part of your process that went into chaos, and you can pull it back by climbing the ladder from complexity to complicated to clear. Okay, that's great theory. And now you're going to be thinking about this all of Thanksgiving, and over your virtual calls with family, you're going to be talking about this over turkey because you saw these little characters climbing a mountain and you can't get it out of your head. That's great. We've done our job. But here are some areas where it actually rubber meets the road with FDA. So places where FDA and industry are actually identifying complexity and putting procedures in place to deal with it. The first one we'll talk about is FDA design controls. It's actually a method of dealing with complexity, of climbing those ladders. Another place is in the realm of FDA submissions. Believe it or not, FDA is doing a lot of work to try to make this a simpler process, and the fundamental thing is they're recognizing that it's not. They're recognizing that it's not a simple domain and doing what they can to push back in the direction of making it a simple domain. The world of biocompatibility is a place where complexity exists and steps have been taken to reduce that complexity or at least help identify it. And in the world of usability and human factors is another example. So let's work through each of these. In the first one, FDA design controls. When you're marching through design controls under stage gate review, developing your medical product, you'll go through at least 4 stages that are very familiar to many: design inputs, design outputs, verification, and validation. And of course, the first question everyone asks is, what do I have to do to satisfy FDA? And FDA's response is, You need to tell us what you need to do based on what your intended use is. And you have to document that in design inputs. You have to realize it in design outputs. You have to test it in verification and validation. Well, what is this other than a method of categorizing unknown unknowns, known unknowns, finding solutions to them, and working your way up to a simple and clear domain where you can put it in your extended quality system and you can run this thing as a company providing a safe and efficacious product. But you didn't think about design inputs that way. It's actually the Cynefin framework in a different guise. FDA pre-submission. If you've been involved in medical device development, you know that the agency has been doing a much better job of taking your calls and letters before your 510K submission, before your de novo submission. Back in 1976, the agency, I think, maybe thought that things would be well ordered if they just made a bunch of bins to place medical devices into. That's what the legislation from 1976 assumes, that there are simple bins and straightforward demonstration of substantial equivalence, which is reasonably well documented in the legislation. But as anyone knows who's been part of this process, it's not as simple as that. And there's been an increasing recognition of the inherent complexity of the process. Even though the categories exist and the product codes exist, even though substantial equivalence has a definition, a legally defined definition, how you place your product in those bins is hard to agree upon. It's much more complex. And pre-submissions are a method of analyzing and identifying which complexity domain you're in. So pre-submissions are a method of climbing those ladders and actually just identifying where you are and what ladders you have to climb. So that's another example of complexity reducing mechanisms that the agency is working on. They've had to deal with this in a number of areas: emergency use authorizations, enforcement discretion, the case for quality program, software as a medical device program, and post-market surveillance efforts that they often place on devices, including PMAs. So they're no stranger to this idea that there's more complexity than meets the eye. But let's go over to industry for just a minute. In the world of biocompatibility, the 2019 version of 10993, the biocompatibility standard, requires what's called a biological risk assessment. It used to be that people would just test everything, and the agency would say, have you tested everything? But this new approach, inherent in it, is a recognition that some materials are simpler and better understood. Some materials are less well understood for a particular application. And so the BRA forces you to do this analysis. What are the known unknowns? What are the unknown unknowns? What are the known knowns? What can we say about this material for this application? And it forces us into identifying that. So you're seeing industry understanding and identifying that complexity exists and putting mechanisms in place to force people to try to address that complexity rather than glossing over it. In the world of usability or human factors engineering, these things exist in FDA guidance. They also exist in the 62366 standard, which is accepted by FDA. What will often happen is a device will be developed according to a particular user's preferences, but the use turns out to be completely non-intuitive for the average user. FDA has become aware of this, and so they put a big focus on usability engineering, which is really just an identification of the unknown unknowns, saying, have you put this into users' hands? Can they use it? Let's see. We don't believe you that it necessarily will be usable. And this has been it's a very challenging area, usability, but for our discussion today, for the purposes of Cynefin, it's another example of a place where complexity recognition, identification, and analysis is tremendously important. I want to give you just a couple more examples, and then we'll open it for questions. One is a device that we saw recently for development, which has multiple navigation functions. There's one navigation function which has amazing precedent in the world of device development and robust techniques for executing it. The second navigation function has no clinical precedent. So the question, Are we ready for an integrated prototype? Are we in the simple domain? Can we just build it with a good expectation of success? Well, the answer in this case is no, but the steps can be parallel. One you can solve through the best practices of the simple or clear domain. You can just develop that prototype according to the way that people do other navigation systems that are similar, and you're going to have great results. I can almost guarantee it. But the other needs novel or exceptive practices to solve. You have to probe, sense, respond. You got to try something. Is that going to be good enough? No? Okay, what about this? You got to put something out there, watch it fail, so that you know what will succeed. So the solution in a case like this is to develop a minimum viable product for function 1 while finding a clinical precedent for function 2. And this is a great example of a place where people will often gloss over complexity and just say, we've got to develop a prototype. Well, what if part of that prototype is able to be developed, but part isn't? Sometimes you've got to recognize the complexity, bifurcate, and then remerge later. I know many years ago we saw an example where somebody came to us with 4-cavity steel tooling, $100,000 worth of tooling. They had a beautiful design. They had no robust testing demonstrating feasibility. And they had kicked off the tooling with urgency because they had to make product. But the design didn't work, and there was no way to fix that tooling. And so this is another example of a place where you say, I've got to get to manufacturing. But oftentimes, if you haven't done the testing ahead of time, all those manufacturing dollars could well be wasted. And on the academic side of the world, as a last example, one of the things that we'll often see is people will come to us and say, I've got a test designed so that I can go to FDA. But the test is more designed based on what the academic is interested in, not on what FDA has actually asked for. It's not driven by safety, it's not driven by efficacy, it's not driven by substantial equivalence. And this is a huge reason for part of the pre-sub program at FDA, is to recognize the complexity, the questions that are going to be asked, and then to develop answers, not to whatever questions we think are important, but to the questions that the agency thinks are important. So in summary, all projects involve complexity. Cynefin is a useful framework for identifying and categorizing complexity. Admitting complexity doesn't mean chaos. It doesn't mean accepting chaos. The fact that you say that there's a mountain to climb does not mean that you're accepting the chaotic domain. You just have to recognize that accepting the complexity that's there and some form of probing is usually going to be required. For this segment, I want to thank my colleague Aaron Kerr, who in our organization has done an awful lot of the legwork around Cynefin and the application of the philosophical thinking to our day-to-day life. But I want to ask you, if you have any questions about this, we'd be happy to answer them now. How does this apply to your program? How does this apply to your project? How does this apply to your life? The identification and categorization of complexity. If you have any questions or comments, feel free to raise your hand. We'll give some time right now for that before we get on to our next segment. Our friend Ken Spencer is live with us.
Yeah, so very interesting, Dean, and the graphics were great, by the way. So, but in terms of this complexity issue and actually focusing on design controls from the FDA, we have so many projects coming forward with machine learning, not just at U of M, but at Stanford and MIT and other places. And it is really frustrating to discuss these things with the FDA. They, even though they know the complexity issue that's there and the elements and things that machine learning can take, they are not equipped to help us get through this thing. I mean, the questions they ask are, I don't want to say naive, but I mean, they just have not put any effort into this. And machine learning, in our opinion, and everybody across the country, there's so many benefits coming down on this thing. There's just not a priority based on that.
Yeah, it's an interesting point you bring up, Ken, and it's a place where what you're probably running into has a lot to do with the usual ways that FDA looks at safety, efficacy, and substantial equivalence. you need to be able to paint a line around the domain and make sure that everything is inside of that line. And so when they hear about a device that can go into sort of an infinite number of states and is going to change its behavior based on things, it completely defies all the classical ways of regulatory science, because they want to be able to draw a line around it and say, okay, is it going to stay in here? I can't help but wonder if, and it may be a while before the solution exists in a regulatory space, I can't help but wonder if some of the solution is what we would call post-market surveillance. I think there's a lot of opportunity to look at things with a provisional clearance, and then to look at them again and to look at them again and to see if the behavior you're getting really merits the original clearance. I remember being in a session where Jeff Shuren, who's the head of Center for Devices and Radiological Health, made the comment. And he's often bound by what the legislation actually says, and he does his best, but he can't get outside of that. He once said, every device we clear to the market is an investigational device, meaning every device we clear is worthy of downstream scrutiny. We can't prove everything is perfectly safe and efficacious. But when a reviewer is looking at it and they say, oh, I can't bound these domains because the machine learning could take it outside of these domains, that's where the rubber is hitting the road. And to your point, not getting some good solutions in the short term. Have you had any inklings, Ken, of discussions and downstream solutions on the horizon, or is this pretty much just a roadblock for projects you've seen?
Well, it's so far been a roadblock. I mean, I think your idea of having provisional things and be able to look back. I mean, many of these machine learning things are. Let's take, for example, Fifth Eye that you've had on there. It's not a decision support system. It is an advisory system, right? And many of the things that are out there right now are advisory in nature. They're not meant to be decision support, and we just cannot get through the FDA to even give a provisional thing to be able to look back, and look at these things. Despite all of the, like in Fifth Eye's case, for instance, we have all of this previous data of people, that we can apply the Fifth Eye approach to and see that deterioration actually occurred many, many hours before it was called. And they just are not buying it. So it's very difficult especially on even the software side, let alone the device side of things that are going on.
That's a great point. I'm sorry for the troubles there, but it's a really important point that you're bringing up. Makes me wonder if some of the folks at FDA who work on that side, it'd be a great conversation to have with some of them on a non-project specific basis. How are we going to address these things?
Yes.
Great, Ken. Thank you for that. We appreciate that feedback and that interaction. Other questions, other comments on this idea of complexity in projects? And if your comment is outside the realm of medical device, that's fine, because a lot of times you're working on something and you say, I get it. There's complexity here that I haven't identified. And then you do that safe to fail experiment. You say, well, let me try this. It doesn't have to work, but let me try this. Oh, look, that cleared up my confusion. If you're having that kind of an experience, we'd love to hear from you. And we'll leave that open for just a minute before we go on to our next segment. But not seeing any more, I want to thank Ken for his comment. And next up, I want to welcome our friend Nikki Kennedy, Dr. Nikki Kennedy, back to the show. Nikki, are you with us?
Hey, Gene, how are you doing?
Doing well. Welcome back.
Good. Thank you. Thank you. I'm working on video. I'm being assaulted by my pets right now. 1 second.
That's fine.
There we go.
Dr. Nikki Kennedy, it's good to have you back. Nikki and I do many times a segment we call From the Front Lines. We're going to pick that back up most likely in January. But we thought before Thanksgiving, we would end this on a high note. So we wanted to bring Nikki back and just do a little bit of a retrospective here and think about where we are and where this is going. Obviously, last week we had Dr. Phil Levy on the show, and he gave us a lot of things to think about, Nikki. He was, and I think you were there telling us about vaccines and some of the things we can expect, B cell and T cell immunity, some of the things that we maybe can't necessarily expect, which is the lingering um, resistance to the bug, just a better ability to fight it. We talked about some of the numbers in Michigan and where things are going. And I think there is this sense as we go into the holidays, which can often be a hard time for people. Um, Boy, what hope do we have here technically from this perspective? And you offered a perspective up which relates back to the days of the HIV crisis, which I thought would make a wonderful high note to go out on before we all take next week off for Thanksgiving. So I wonder if you could start to unpack those thoughts for us just a little bit.
Well, I will do my best because I will say I am not an HIV researcher by any means, but I remember, and I know Jean and I were talking earlier, and Jean, I had mentioned to you that I was in high school when the HIV crisis was still quite new and before things had really started to blossom on the research side. And there was a lot of fear. I remember specifically, and I had shared with you that when I was in high school, I had to do a debate for English class. And I'd like to say I'm happy I won that debate, but I'm not really sure if I am, because it was whether or not children with AIDS should be allowed in school. There was the controversy of, I believe it was a young Ryan White who ended up passing of HIV. I think he had gotten it from a blood transfusion from a disorder he had when he was young. And I was assigned the con side, why children with HIV should not be allowed in school. And I think we've come so far since then, but at that time, I was able to find a lot of compelling evidence to not let these kids be out with their peers. So I think when we look back at that time frame, we see how much has changed. We see the science has evolved. I actually took the liberty, Jean, and I don't know if you want to share this with your guest or not, but if you look at the HIV.gov website, they have a great timeline summary of the entire HIV AIDS crisis and all the research that came out, going all the way back to 1981 when they first noticed a pattern of, I think it's pneumocystis pneumonia, as well as Kaposi's sarcoma. And they were in opposite sides of the country. I think the pneumocystis outbreak they were publishing about in LA, whereas the Kaposi's was in New York, all about the same time. So from these isolated outbreaks of very strange findings, researchers over less than 40 years have put this together, realized that they were related, figured out the science behind it, actually went through and did a bunch of research, basically invented human genomics as a huge science in medical device and pharmaceutical development, which really we didn't have before the 90s. And I believe, if I recall, just last spring when we were first hearing about the COVID crisis blossoming, I think it got a little buried, but some researchers in the UK announced that they had just found a cure for HIV. I went to take a look at this and I actually saw that some of our U of M researchers are involved with this too. They published as recently as September as they found a molecular antibiotic that will actually eliminate T cells that are infected with the HIV virus. So for me, that is such an amazing and wonderful thing because HIV is a slippery player, just like COVID. We didn't know what it was. It was not something that was affecting a lot of people, but it was affecting enough people. And again, this is a group of people who I like to think are certainly well respected and empowered these days, but back in the 80s, that may not have been as true. And oftentimes maybe didn't get the same attention to their medical needs that they should have deserved. So trying to piece all that together, again, not even having the computing power that we had back in the 80s, and to, in less than the lifetime of, maybe a millennial, be able to solve this, I think that's pretty awesome. And I know it's difficult right now. People are upset. Things are getting shut down here in Michigan with COVID again. People don't know what's going on. You hear all these news about vaccines. I really enjoyed Dr. Levy's talk last week. I thought he just brought a great spotlight on a lot of things that people don't often talk about. But I think that we need to continue to think in a forward direction. The amount of time and effort that's being spent on researching the human immune system, which is something we don't know a whole lot about really up until now. I think this is going to open a lot of doors going forward. Maybe not today, maybe not tomorrow, but 5 years, 10 years from now, we're going to be having a whole slew of new technology and research that will help us treat autoimmune disorders that are just devastating, cancers that are devastating. It's coming. I think that this is just the herald of a new age, the same way that HIV was for us about 25 years ago or so. I hope.
I think it's a really encouraging perspective. And when you and I were chatting, that's where I wanted to sort of leave our show before the Thanksgiving holidays. Everybody goes off and thinks about their family and thinks about life, and hopefully has a wonderful Thanksgiving. That we've seen these kinds of things before, maybe not at this scale, but you don't hear as many people dying of HIV and AIDS as you used to. And you bring in some light to the work that's been done, and the work that's being done on COVID is immense, and
Yeah.
just to give that the time that it needs, because we're going to see some good things happening here. Dr. Nikki Kennedy, in addition to her practice and all of her credentials, runs Arbor Hive, and you can get to Arbor Hive at arborhive.com, or you've got a podcast yourself, arborhive.podbean.com, specifically focused on getting physician innovators into the process. And we just love what you're doing, Nikki, and it's always great to talk to you. And I think with that, maybe Nikki and I will both close it off together and just thank you so much for being with us here on MedTech Crossroads. We're so glad. No show next week. Enjoy your families. Enjoy Thanksgiving with whoever you can be with. And we will see you the week after, back to our regular schedule with guests. So we're looking forward to it. Thanks, Nikki. Thank you all.